So, welcome back for the part 3 of our series where we explore the roles of glucose transporters and enzymatic activity in detail. So, let me give you the outline for this part 3. Number 1, kinetics of enzymes and glucose transport and how enzymes functions and glucose movement are regulated dynamically. So number 2, we'll discuss various types of glucose transporter protein, the so-called GLUT or GLUTs. Then we go and discuss isoenzymes like glucokinase and hexokinase and their important roles in glucose metabolism. And lastly, clinical significance of glucose transports and enzymes activity. So let's begin or start with enzymes kinetic. So the role of enzymes is to speed up chemical reaction by binding to substrates and forming a complex. When you create a graph with substrate concentration on the x-axis and reaction velocity on the y-axis, as the amount of substrate increases, the reaction rate rises until all enzymes active sites are filled up, a point called saturation, which correspond to the maximum reaction rate or the Vmax. This means that increasing the substrate concentration further won't speed up the reaction because the enzymes is already fully saturated. Km, by the way, stands for Michaelis constant, is the substrate concentration at which the reaction rate is half uh, of Vmax and reflect the enzyme's affinity for its substrate. A low Km means high affinity, while a high Km indicates lower affinity. These two values, Km and Vmax, show how enzymes adjust their activity according to substrate level. For example, glucokinase found mainly in the liver and pancreas has high Km and Vmax, becoming active when glucose levels are high, such as after meals, to help regulate blood sugar and insulin. In contrast, hexokinase, present in the skeletal muscles and adipose tissue, has a low Km and Vmax, allowing it to efficiently phosphorylate glucose even at a low concentration, ensuring a steady energy supply. So let's take another look at the Km values of these two key enzymes. Glucokinase has a Km of 5 millimolar, while hexokinases Km value is 0.05 millimolars, meaning glucokinase Km is 100 times higher. This higher Km enables glucokinase to become active only when blood glucose levels are elevated, allowing pancreatic beta cell to respond appropriately by releasing insulin to maintain glucose balance. So, what are the physiologic importance of these two key enzymes in glucose metabolism? Now, hexokinase and glucokinase play distinct roles in glucose metabolism, tailored to their tissue distribution and kinetic properties. Hexokinase, found in most tissues, has a low Km and is active even when the glucose is scarce, ensuring vital organs like the brain, nerves, the red blood cell, etc. receive energy during fasting. On the other hand, glucokinase, located in the liver and the pancreatic beta cells, has a high Km and only becomes active when glucose is abundant, such as after a meal. This allows the liver to store excess glucose and the pancreas to sense rising blood sugar levels for insulin release. Together, they balance glucose utilization across fed and fasting state. Now, I often use an analogy for this. Glucokinase only eats when the table is overflowing with food, but when your sources are limited, it steps back and allow other vital organs to take up the glucose first. Now, glucose transporters also operate according to kinetic principles defined by Km and Vmax, with each transporter type possessing a unique Km value. These differences are shown on this slide. GLUT1 and GLUT3 are high-affinity, insulin-independent glucose transporter that ensures steady glucose supply to vital tissues. GLUT1 is widespread, supporting basal glucose uptake especially at the blood-brain barrier and red blood cells. GLUT3 is primarily found in neurons with an even higher affinity to efficiently meet the brain's high energy demand. Both are essential for maintaining constant glucose availability regardless of insulin level. GLUT2 is a low affinity, high capacity glucose transporter that acts as a sensor in the liver as well as in the pancreas, detecting high blood glucose level to regulate insulin release and glucose storage. In contrast, GLUT4 is insulin dependent and primarily found in muscles and adipose tissue, where it facilitates glucose uptake after meals to support energy storage and use. Together, they coordinate glucose regulation based on the body's metabolic state. The clinical tie-in, like GLUT1 deficiency, can cause low brain glucose, leading to seizures and developmental issues. With GLUT4, problems reduce insulin-driven glucose uptake, causing insulin resistance and higher blood sugar. Now, in part 2 of the series, we covered the SGLT co-transporter within the context of active transport. Now we will move on to examining its kinetics. As illustrated here nicely, the kidney's glucose reabsorption involve two co-transporters with different affinities. SGLT2, which has a high Km, and SGLT1 has a low Km. Because of this, roughly 90% of glucose is reabsorbed in the early part of the proximal tubules where SGLT2 is present, while only about 10% is reabsorbed later by the SGLT1 in the distal section of the proximal tubules. Now we go back to GLUT4. GLUT4 is a unique glucose transporter found in the skeletal muscles and the adipose tissue. It is characterized by a moderate affinity with a Km of about 5 millimolar, unlike most other glucose transporters. GLUT4 requires insulin to function. When insulin is available, it triggers GLUT4 to relocate to the cell membrane, enabling glucose to enter the cell where it can be used for energy or stored. This insulin dependence set GLUT4 apart from other glucose transporter that operates independently of insulin. So all glucose transporters, the GLUTs, are located on the cell surface except for GLUT4, which is usually stored inside the cell until activated. As shown here in the skeletal muscle, insulin signal causes GLUT4, which is normally stored inside the cell, to move to the cell surface, which is termed as translocation, where it can take up or pick up glucose on the surface. So the clinical application of relevance for the isoenzymes in fasting, hexokinase dominates to preserve brain and muscle function. In fed state, glucokinase buffers excess glucose, preventing spikes and aiding insulin secretion. In MODY2, or maturity onset diabetes of the young type 2, is a mild form of diabetes caused by mutation in the glucokinase gene, which raises the threshold for insulin release. This leads to stable, mild elevated blood glucose level from a young age, usually without symptoms or serious complication. Now, the treatment is often unnecessary as the condition rarely progresses or causes damage. So let's go through some exercises examining how enzymes kinetics vary across different glycemic conditions, albeit some of these scenarios might not be physiologically accurate. They will help us better understand the mechanism behind different kinetics behavior. Starting with both glucokinase and hexokinase, Glucokinase normally has a high Km and high Vmax, while hexokinase has a low Km and a low Vmax. This typical profile support normal blood glucose level, the top two levels. Now, if we change glucokinase to have a low Km instead of a high one without altering Vmax, what would happen to blood glucose? It will result in hypoglycemia since it will trigger insulin release at a lower blood glucose level, causing the body to lower blood sugar excessively and potentially leading to hypoglycemia. Next, lowering Vmax of glucokinase while maintaining the same Km would likely cause hyperglycemia. This happened because the enzyme's ability to process glucose is diminished, which in turn reduces insulin release and allow blood glucose level to increase. Now, if hexokinase Km increases while Vmax stays the same, its affinity for glucose decreases, meaning it requires higher glucose level to work effectively. This reduces glucose metabolism in the cell, causing blood sugar to rise and leading to hyperglycemia. And finally, elevated Vmax of hexokinase with normal Km would generally result in increased glucose utilization by tissues, leading to lowering blood glucose level or causing hypoglycemia. So that concludes part 3. Up next is part 4, where we'll explore how glucose is converted into ATP.